virus, T2 virus, is DNA. So if I had to define a virus, I would say a virus is, you're rocking the camera. It was rocking. Not a big deal. A definition of a virus would be it's nucleic acid surrounded by protein. And that's an oversimplified definition, but basically that's what it is. Viruses would be nucleic acid surrounded by protein. And this structure we're showing here is a T virus. So I want you to look at this experiment and try to understand, how did Hershey and Chase, which could be a good question, how did Hershey and Chase prove that it was the DNA and not the protein coat that's the genetic material of a virus? So they worked with T2 virus, and they worked with the organ of E. coli, and I think the answer is here. What part goes into the bacteria? Not the protein, just the DNA. And so they developed a way to show that the DNA entered the virus. So that may be one of the questions that I will ask. Let me give you a hint to how it works. He used radioactive phosphate, P32, and he used a radioactive sulfur, S35. P32, S35. Looking at the structure of DNA, what would be the elements that you find in DNA? What is it? That's a compound. Give me the elements. Probably carbon. What else? Nitrogen. Hydrogen. Oxygen. In proteins, you pretty much say the same thing, right? What would you find in DNA that you will not find in protein? No, you have an NH2 in proteins, in amino acids. You have phosphorus. Remember sugar phosphate backbone? So you have a lot of phosphate in DNA. So if you use a label P32, it's going to label DNA. And there are like three amino acids that are high in sulfur. And for the most part, they're pretty much found in most proteins. And cysteine is one of those. And as you were studying proteins, I think you may have talked about disulfide bridge. Methionine is another one. So there are several amino acids, but there's no sulfur in DNA. So using radioactive sulfur, you can distinguish how this works. So I would like for you to read through this and kind of go to see how they differ. How did he do an experiment using radioactive phosphate and radioactive sulfur to distinguish that it's the DNA that enters the cell and not the protein. Although we clearly show it here, but we have to have an experimental design to show this. And unlike this slide may indicate, he can't use them both at the same time. You have to do an experiment with the sulfur, find out what happens, and has to do an experiment with phosphate, radioactive phosphate, to see what happens. And to answer the question conclusively, in a virus, what is the genetic material? So, let's move on. This is a plant virus called tobacco mosaic virus. And this is an RNA virus. Now remember, I had already defined a virus as a protein coat. In a tobacco mosaic virus, there are actually two kinds of protein coats. So you can have a tobacco mosaic virus that displays one protein code and another tobacco mosaic virus that display another one. He has them displayed here, purple and blue. Is that okay? One virus, tobacco mosaic virus A, has a kind of purple protein surrounding it. Tobacco mosaic virus B has a blue protein, and this is only a cartoon to demonstrate a point. The protein's not really blue and purple. It's just showing that there are two different types. At any rate, the question is asked, if you put the protein from virus B around A, then allow it to infect the tobacco leaf, what kind of protein coat will virus A produce at the end of the experiment? And the opposite for virus B. So, in other words, use protein A and let it associate with virus B, the RNA B. What will happen if it replicates itself? Will it continue to produce the same protein that's associated with? And now, obviously, the answer is RNA A will produce an RNA A protein, and RNA B will produce an RNA B protein. So this is an actual experiment that was performed to show that the genetic material of an RNA virus is the RNA, not the protein. The protein does not determine the RNA, but it's the RNA that determines the protein. Okay, now the good stuff. Looking at the structure of DNA, what are the bases that you find in DNA? Right, and so RNA substitutes uracil with thymine. And we also classify them as purines and pyrimidines. Just at a glance, can you tell me the difference between purines and pyrimidines? What is the difference between, looking at the diagram?
Double carbon bond.
One has a double ring, right. Another one that has a single ring. That smaller ring is called an indole ring. So purines have an indole ring. Pyrimidine is only one ring with 6 carbons. So this indole ring is part. So you can look at the chemistry and tell the purine from the pyrimidine. These are purines, adenine and guanine, and the pyrimidines, of course, are cytosine and thymine. And uracil, which is found in RNA, is a pyrimidine. And the rings are numbered. The carbons, the elements in the ring are numbered, 1 through 6, and then in the pyrimidines goes to 9. And the purines are just 1 through 6. And then we have the ribose sugar and deoxyribose sugar. So what is the difference between the ribose sugar and the deoxyribose sugar? Oxygen. Obviously, deoxy means it's missing an oxygen. But at what point? 2' is missing an oxygen. So why do I use prime when I'm talking about the sugar, and I don't use prime when I'm talking about the bases? So if I use prime, you know I'm talking about the sugar. If I say carbon 1', it would be the sugar at carbon 1. If I say carbon 1 here, it would be in the base, where actually it's a nitrogen. In all of them, actually. So at the position 1 in the basis, it's just simply 1 through 9 or 1 through 6. And here, 1', we're talking about